Welcome, everybody. Good afternoon. Thanks very much for attending, for connecting to this first virtual digital liver seminar. As you know, the current situation made us to change our conventional way to do this kind of seminars in face-to-face to this new form that necessarily we must adapt, that is the digital. Anyway, for us, it's a real pleasure. For me, it's an honor that Laura Fouassière is here today. Laura accepted our invitation, thinking that she could visit us in Barcelona. It was not possible because the seminar was planned for March, if I remember well, and it was not possible to do it. So now she starts, she is our first invited panelist in this new series of liver seminars. The way to present and the way to make the discussion is new for the liver seminars, but I am afraid that it becomes being very familiar for all of us because it has been the only way to interact in the present situation. So Laura made the PhD in Paris, in Sorbonne University, but after the PhD, she spent almost 6 years or more, a little more than 6 years in the US, first as a research trainee at the UCSF in San Francisco, in the laboratory of Mina Wiesel, and later on as postdoctoral senior in Denver, Colorado, with Drs. Fish and Doctor. And she was involved in liver fibrogenesis and in the second part in biliary physiopathology. So in 2002, she came back to Paris, to the Saint Antoine Research Center, and in 2005, 3 years later, she completed the competitive, or she was successful in the competitive exam for a permanent research position. So she has, of course, her own group from this time, which is managing a particularly research line focused on biliary physiology in pathophysiology. The special focus of LOGA is EGF, the epidermal growth factor receptor pathway, the involvement of EGF receptor in EMT, in epithelial mesenchymal transition, in cancer-associated fibroblast, or resistance mechanisms and anti-cancer therapy, but particularly in cholangiocarcinoma. In fact, she is co-founder of the European Network for the Study of Cholangiocarcinoma, and she is participant in the management committee of the ECOAST, the ECOAST that was created from the European Cholangiocarcinoma Network. Is a referent in the cholangiocarcinoma research, and is for many of us the requirement. The specific way to better understand the cholangio, her works and her recent excellent reviews. Laura is author, PI author of many excellent original articles in relevant journals in the field, pathology, clinical cancer research, and so on. And her group has excellent recent reviews in the area. So, Laura, for us is a real pleasure to have you here. Thanks very much for accepting our invitation, and it's all for you right now. Thank you.
Thank you very much, Isabel. So the outline of my presentation will be the following. So I will introduce briefly cholangiocarcinoma and EGFR. And then I will talk about the role of EGFR in epithelial mesenchymal transition. And then I will present some work regarding the regulation of EGFR signaling by the scaffold protein EBP50 and by the tumor microenvironment, more specifically by the cancer-associated fibroblasts. And then I will finish by this work that was recently published on resistance mechanism to anti-EGFR in CCR. If I present this work today, is because I had the chance to work with very talented and wonderful postdoc researcher, Audrey Clapeyron, and for the first part of my presentation, and of course, and Javier Vaquero. And they are both now PI. Audrey is PI now in a big pharma company in France, and Javier is now PI here. I mean, here, not in Paris, but in Barcelona. So I'm very proud of them. So cholangiocarcinoma is a cancer of the biliary tree. And it can be divided in 3 subtypes according its anatomical site of origin: intrahepatic cholangiocarcinoma, perihilar cholangiocarcinoma, and distal cholangiocarcinoma. This cancer has poor prognosis because it's usually diagnosed at advanced stages. It has this main characteristic that it has a prominent stroma compartment with high content of cancer-associated fibroblasts. And we can easily recognize those CAF with alpha-SMA immunostaining here. The therapeutic options are very limited. There is one curative therapeutic option, that is the tumor resection. And otherwise, this is a palliative chemotherapy associating gemcitabine, and cisplatin. As many other cancers, of course, it's not particular to cholangiocarcinoma, there are many signaling and molecular networks that are involved in cholangiocarcinogenesis and progression of cholangiocarcinoma. And one of these signaling pathway we have been interested in since many years is EGFR. So EGFR is a tyrosine kinase receptor, and upon stimulus, it activates 3 major intracellular signaling pathways: STAT, ERK, AKT, leading tumor proliferation and progression. What we know, excuse me, what we know about EGFR in CCR is that EGFR is highly expressed in CCR, mainly at the plasma membrane. And a very nice study from Llovet's group in Barcelona has identified a molecular signature of EGFR in intrahepatic CCR. It's the expression of EGFR is associated with tumor progression and poor prognosis. And it's also a risk factor of tumor recurrence in intrahepatic CCR. If we go to the laboratory and we do some experimental manipulation using human CCR cell lines, it is possible to activate this pathway with EGF, and upon EGF stimulation, here this is a Western blot, showing that the activation status, here the phosphorylation form of EGFR, is activated, that EGFR is indeed activated upon stimulation with EGF, and also that the 3 pathways are JAK-STAT and RAS and AKT are also activated upon EGF. If we check carefully the cells, what we have observed, so the cells have been starved for overnight. So in absence, oh, sorry. Oh, my movie doesn't work. You can see that the cells stay in cluster and they don't move much. But if you add EGF, the cells are getting to separate from each other and to move. So meaning that EGF induces cell scattering and motility in CCR cells. This phenotype is mediated by EGFR. If you add an inhibitor of EGFR, you block the cell scattering. And this is also mediated through two intracellular signaling pathways, ERK and STAT3, because both inhibitors of both pathways inhibit the scattering phenotypes. So from this, we wonder if and this is specific of EGF because we try other ligands of other tyrosine kinase receptors and we didn't see any scattering phenotype upon PDGF or VEGF. So from this first observation, we thought that maybe EGF-EGFR axis was involved in epithelial mesenchymal transitions. So I'm going just here to introduce briefly what is epithelial mesenchymal transition and to present some steps of this process. So EMT is an embryonic biological process by which the epithelial cells are going to lose gradually their epithelial phenotype to gain mesenchymal traits. And in cancer, EMT is well known now, contribute to metastatic dissemination of tumor cells. What are the main steps? So upon EMT stimuli, there is an increase of EMT, what we call EMT inducing transcription factors, such as SNEL, SLUG, ZEB1. And those transcription factors are very important because they are going to downregulate epithelial markers, such as ikadorin, and other epithelial markers. And you are going to lose the junction between cells and to lose apico-basal polarity. Of course, those transcription factors are involved also in the expression of mesenchymal markers, leading migration, ultimately migration in vision of the cells, tumor cells. EGF disrupt cell junctions, that we have observed this, and they disrupt adherence junction. As you can see here, upon EGF, the ikadorin is not localized anymore at the plasma membrane, like I'm going to use the pointer. And like beta-catenin, so you don't see anymore both protein at the cell-cell junction. And what we have shown also that EGF induces mesenchymal markers expression and cell invasion. Here you can see upon EGF, there is an increase of SLUG, ZEB1 expression, N-cadherin expression, and alpha-SMA expression. And all these features are inhibited with gefitinib, an anti-EGFR. And that lead ultimately an increase of invasion, cell invasion. So this work has been published a few years ago in this publication. Now I'm going to talk about the regulation of EGFR. We have performed two main studies, one showing that EGFR can be regulated at the intracellular level by the scaffold protein EBP50, and another study that showing that EGFR can be regulated at the extracellular level by the tumor microenvironment, in particular by CAF, cancer-associated fibroblasts. So I'm going to introduce briefly EBP50. So EBP50 is a scaffold protein. It has two PDZ domains. Those are protein-protein interaction domains. And his main function is to organize functional complexes and facilitate kinase substrate interaction, leading accurate coordination of signaling pathway. I want just to precise that EBP50 is a cytoplasmic cytoplasmic protein, and it's concentrated beneath the plasma membrane. In liver, here, this is human healthy liver. On the left, you have a PCR. And in liver, EBP50 is highly expressed in hepatocyte, in bile duct, and in epithelial cells isolated from gallbladder. Its localization is mainly concentrated, the protein is mainly concentrated at the canalicular membrane and at the apical membrane of cholangiocytes. When we started this work, what we knew it was that EBP50 was capable to interact with EGFR. It was already published. But what we didn't know at that time was the functional role of this interaction. So we started to perform some studies, in vitro studies first, using a very differentiated CCR cell lines in which EGFR is expressed at the plasma membrane. EBP is also expressed at the plasma membrane. They both colocalize with E-cadherin, and they both, here this is a co-immunoprecipitation showing that they both interact to each other. The strategy we decided to use at that time was to downregulate EBP50 by siRNA, as you can see here, in CCR cells. We first look at the cell signaling. Here, the Western I showed you before, I used the same. And what we were able to show, it was that when the EBP is downregulated, there is a higher basal activation of EGFR, and also higher basal activation of the some intracellular signaling pathway, but also higher activation of EGFR upon stimulations. So we decided to go further to understand why. So the first hypothesis was, well, maybe the cells express more since EBP is not anymore in the cells. Maybe EGFR is more expressed at the plasma membrane. And it was the case. This is a flow cytometric analysis showing that in basal condition, when EBP is downregulated, there is higher expression of EGFR at the cell surfaces. Of course, in control cells, when you add EGF, there is a downregulation. The receptor is internalized, as it's known. But still, in cells downregulated with EBP50, you have still a higher expression of EGFR at the cell surfaces, at the cell surface. So meaning that EBP is a negative, in fact, regulator of EGFR. Of course, we looked precisely the phenotype of the cells. And when we checked the cells, what we were able to see was this. So the cells are scattered. They have a fibroblastic-like phenotype, meaning that probably they were undergoing EMT. And it was the case because in absence of EBP50, we were able to demonstrate that loss of EBP50 induced EMT. It induced loss of E-cadherin beta-catenin at the plasma membrane. It induced the expression of mesenchymal markers and cell migration. And all these features were inhibited with anti-EGFR, indicating that indeed loss of EBP50 upregulate EGFR signaling, inducing EMT phenotype. The next step was to verify, to check whether this EBP-EGFR interaction is maintained or not in human CCR samples. So we investigated by immunohistochemistry the expression of EBP50 in CCR cells, in human cholangiocarcinoma samples. We found that TBP50 is expressed in most of the case. And as you can see, it was expressed mostly in the cytoplasm of the cells. So it's not anymore concentrated under the plasma membrane. And this localization, cytoplasmic localization, is associated with EGFR expression, EGFR that remains at the plasma membrane. So meaning that, suggesting that probably most of the interaction between EBP50 and EGFR is lost in cholangiocarcinoma. And this work has been published a few years ago, and we were able to conclude that loss of EBP50 stimulates EGFR activity to induce EMT phenotype feature in cholangiocarcinoma cells. The next work we did regarding the regulation of EGFR was whether it could be regulated by the stroma cells. And of course, we focus on CAF, and I'm going to explain why. As I said before, cholangiocarcinoma is mainly characterized by a prominent fibrous stroma and by the presence of alpha-SMA positive cells that characterize the CAF populations. It has been shown that CAF in CCR is correlated with shorter overall survival and disease-free survival rate. And at that time, when we started the study, it was shown in other types of cancer that CAF were able to produce and to secrete EGFR ligands, such as HBEGF. So my idea was to check whether CAF from cholangiocarcinoma samples express HBEGF. So we started by some immunohistochemistry studies in human samples, where we show here an expression of alpha-SMA, an expression here of EGFR that is mainly expressed in tumor cells here, but also we were able to show an expression of HBEGF in the fibroblastic-like cells in CAF. So from this, we tested whether liver myofibroblasts contribute to cholangiocarcinoma progression through EGFR signaling. And we first performed this experiment in vivo experiments. So this is the model, the experimental model we used for CCR in mice was the subcutaneous xenograft model, in which we co-injected human CCR cell lines with human liver myofibroblasts. And in the second group, we injected only CCR cells, and we treated not the cells, but the mice with anti-EGFR gefitinib. And what we were able to see was that liver myofibroblasts promote, here in gray, promote tumor growth, compared here to the CCR cell alone. You can see here the tumor when both cell types were co-injected. And then this was mediated through EGFR pathway, because after treatment with gefitinib, the tumor growth was much lower. You can see here the tumor size. We check the EGFR signaling. We showed that higher EGFR signaling in tumor that were both injected coming from CCR cells and myofibroblasts. And this signaling was indeed, in fact, inhibited in mice treated with gefitinib. Of course, we also performed some other analysis. I'm going to show two of them here. We check the tumor proliferation and the angiogenesis. And as you can see, when both cell types were co-injected, we were able to see higher proliferation rate in tumor cells, a higher number of vessels. Here they are staying with CD31 antibody. This is the quantification, higher proliferation, higher number of blood vessels, and both features were inhibited when the mice were treated with gefitinib. And then we decided to understand a little bit more about this communication between myofibroblasts and CCR cells. So we performed some in vitro experiments, and the model we used was this one. I'm going to present the model. We prepare some conditioned media from human liver myofibroblasts, and this conditioned medium was added to CCR cells. The first experiment we did was to verify, to check whether this conditioned media contains HBGF, and we performed some ELISA, and we found some HBGF secretion in this conditioned media. And when this conditioned media is added to CCR cells, there is an increase of EGFR signaling in CCR cells. Here are 3 different conditioned media prepared from 3 different patients, and EGFR signaling is increased. We were able to see that upon conditioned media, there is a cell scattering and a change in the cell phenotype with a disruption of cell-cell junction, a disruption of cell-cell junction. And these phenotypes was completely blocked when the cells were incubated either with neutralizing antibody against HBGF or against EGFR. The phenotype is completely abolished, or even against an anti-EGFR gefitinib. Also, upon conditioned media, the cells have a higher cell migration rate, and this is inhibited with gefitinib. So, in conclusion, we showed that liver myofibroblasts, or CAF, communicate with CCR cells, with tumor cells from cholangiocarcinoma through an HB-EGF-EGFR axis. Of course, we didn't stop here. We wonder whether this communication was reciprocal. And we found that CCR cells, tumor cells, trigger expression of HBGF in liver myofibroblasts through TGF-beta. That is also involved in myofibroblast activation. Meaning that CAF and tumor cells communicate with each other through HB-EGF-EGFR axis. And this was published a few years ago in Hepatology. Now I'm going to talk about the last part regarding the resistance mechanism to anti-EGFR in CCR. And I will explain why I decided this in a few minutes. So I would like just briefly to say that there are 2 main molecules that are involved in the inhibition that are used to inhibit EGFR, antibodies first, and tyrosine kinase inhibitors. This is not an exhaustive list, but most of them have been tested in human, and none of them worked. So it's not also an exhaustive list, but I'm using this table to show you that just one that didn't work, but it was the one that used erlotinib, and this is the only clinical trial in phase 3 that has been conducted with anti-EGFR. And since any anti-EGFR worked, I was wondering why. So it's for this reason I decided to study the molecular and cellular mechanisms involved in the acquired resistance of CCR cells to erlotinib, the only anti-EGFR that went to clinical phase 3. And for that, we generated in vitro models. We had 2, and those were erlotinib-resistant CCR cells. We generated those cells by incubating 4 different CCR cell lines with escalating doses of erlotinib. And we generated erlotinib resistance pool. And we used these cells to understand, to dissect the molecular mechanisms behind resistance to anti-EGFR. Of course, we validated first our model. EGFR is indeed inhibited in resistance in all cell lines. They are much less sensitive. They are much more resistant to erlotinib here. For the 4 cell lines in black, those are resistant cells. And then we observed, as usual, this is a recommendation I give for all students when they come, look at your cells, because you learn a lot when you look at your cells. And what we observed when we were generating resistant cells, it was this phenotype that is really different from the control parental cells. Parental cells grew in cluster, but the resistant cells were much more different. They had a scattered phenotype, and they were more fibroblastic. We performed some transcriptomic analysis from those cells, and we found that resistance was cells was the transcriptomic analysis of resistant cells was associated with a metastatic signature. Of course, we said, okay, maybe epithelial mesenchymal transition is involved in this resistant phenotype. And it was. I'm going here to show you just one CCR cell lines that show that resistant cells lose icatibant and beta-catenin at the plasma membrane. And in these cells, you have an increase of EMT-inducing transcription factors, such as SEB1, SLUS, and SNED in these cell lines. And there is higher migration of the cells. So meaning that erlotinib-resistant cells, CCR cells, tumor cells, undergo to EMT. Because EMT is now well known, is linked to stemness, we investigated whether the cells had stemness features. And we look at first at this signature, CD44 CD24 low, that is associated with EMT and stemness phenotype in cells. And by cytometry analysis, flow cytometric analysis, we found that there is higher population, CD44 plus CD24 minus, in resistant cells. And this was along to a higher sphere formation unit in resistant cells, meaning that these cells, resistant cells, display stemness properties. But if there is an EMT and stemness phenotypes, what are the alternative signaling pathways that are involved in this phenotype? It's not EGFR because it's blocked, but it's probably another signaling pathway. So we decided to screen by protein urticaria array 49 RTK using this approach, and we discovered that the only tyrosine kinase pathway, tyrosine kinase receptors that were activated, was the insulin receptor were the insulin receptor and the IGF-1R receptors. So they are both activated in resistant cells. Regarding the specific ligands of those receptors, we found that IGF-2 was much highly expressed in resistant cells. We didn't find any expression of insulin as expected, and very low expression of IGF-1R and no change, IGF-1. So the next step was to block those pathways and to see whether we can reverse the resistance phenotype of these cells. And we used two tyrosine kinase inhibitors of insulin receptors and IGF-1R receptors. And we were able to find that when we block insulin receptor, IGF-1R receptor, there is a decrease of cell proliferation of resistant cells and plasticity of those cells, meaning we have a decrease of EMT feature, as you can see. The Lansitime was much more efficient, but you can see a decrease of ZEB1, of slug here and SNEL, and a decrease of the migration of the cells, and also a decrease of sphere formation in the resistant cells. So from this in vitro studies, we went to the in vivo to validate whether Lansitime can have an efficacy in treatment. What we observed that the resistant so we used the same model, the subcutaneous xenograft model of CCR, and we observed that erlotinib resistant cells, CCR cells, showed an increased tumorigenic potential compared to parental cells. What we also observed was that the tumor of resistance had higher tumor stroma content. And you can see here the expression of alpha-SMA, a marker of CAF, that is highly expressed in resistant tumors. I'm going to come back with the stroma later. Now, if we treat the parental tumor with erlotinib, we have a decrease of tumor growth that was expected, and a little decrease, and also a decrease in presence of Lansitime. But now, if we do the same treatment with resistant tumor, with erlotinib, you don't have any effect because they are resistant to erlotinib. But when you treat the mice with Lansitime, you have a complete downregulation of the tumor rate, like in control. So what also that was data that we could observe, and it was very intriguing, was very surprised for us, it was that we found some change in stroma. And here, this is the alpha-SMA immunohistochemistry. You can see that in vehicle in resistant tumor, there is a much higher alpha-SMA immunostainings. Erlotinib do not change much this staining. But Lansitime, you have a complete downregulation of alpha-SMA positive population. So we wonder if we had a link between CAF and insulin receptor IGF-1R signaling. And we check if indeed CAF expressed or not IGF-2 and IGF-1R receptors. And we found that IGF-2 was expressed in CAF, you can see here in resistance, and parental tumors, meaning that probably it was suggesting that CAF produce IGF-2 that contribute to resistance mechanisms to anti-EGFR in tumor cells. We also check whether IGF-2, since myofibroblast, liver myofibroblast express receptors of IGF-2, whether each IGF-2 could have an effect on CAF also through an autocrine pathway. And what we found, and we use here two models of CAF, in vitro model, myofibroblast from liver, and what we found it was that IGF-2 activates insulin receptor and insulin growth factor receptors in both models, and that IGF-2 increased the proliferation of liver myofibroblast, and that lensitinib is able to downregulate the CAF proliferation, the activation, and the extracellular matrix synthesis. The last two results, I don't show you the last two results here. So meaning that IGF-2 has also an effect on the stroma cells, on the tumor microenvironment, when the cells are resistant to erlotinib. And this is a cartoon recapitulating everything, all data we were able to generate. So meaning that after a long-term treatment with erlotinib, there is an increased expression and activation of ER and IGF-1R receptors. And those are, and also an increased production of IGF-2, leading the phosphorylation and activation and progression and activation of EMT, stemness feature, and chemoresistance. But we have also the contribution of stroma cells, and they contribute by producing IGF-2, and they contribute also, by producing IGF-2, but also IGF-2 is able to increase their proliferations, creating a kind of vicious circles. And that both effects can be inhibited by tyrosine kinase inhibitors, again, against ER and IGF-1R receptors. And this work was recently published in Clinical Cancer Research with Javier Vaquero. I'm almost finished. Before to give you the take-home message, I would like just to tell you a few words about our future direction. Currently, we have initiated collaborative work with Jesus Benales, and we are currently investigating the role of cholangiocarcinoma-derived extracellular vesicles containing EGFR in tumor stroma communications. I would like just to give a few words why. First of all, as you know, extracellular vesicles are very important mediators that are very important now in cell-cell communications. So we know that. And why we decided to start this study, it was because Jesus in 2017, in this paper, showed that extracellular vesicle isolated from CCA tumor cells express highly, they have high content of EGFR in their extracellular vesicles. And we check the presence of these receptors, and that in extracellular vesicle of culture cells, and it was indeed highly expressed. So we're now continuing this work, and this work is in progress. We have set up all tools with Javier Vaquero and Ander Arbeláez. And we are currently continuing. Take-home message of my presentation, or what I've showed you, is that we can say that EGFR is a major player of cholangiocarcinoma progression by inducing epithelial mesenchymal transition, by inducing migration and invasion. Also, that EGFR is regulated, highly regulated, intracellularly by the scaffolding protein in a negative way, and also by the tumor microenvironment, by CAF, that produce EGFR ligands. And we have uncovered mechanisms to explain why anti-EGFR do not work in patients, indicating that when EGFR is inhibited, you have a resistance mechanism that go through activation of insulin resistance, IGF-1R, insulin receptors, insulin growth factor 1 receptor, cell signaling that induce EMT and stemness features. And there is also contribution of the tumor microenvironment of CAF here that produce IGF-2. Of course, I would like to thank people who had participated to this work, and Javier, especially Javier Vaquero and Audrey Clapeyron, and all my close collaborators, Linda, and many of other collaborators from France, from my institute, or international collaborator I currently have. And that's it. Thank you very much for your attention. Those are both interesting questions. For the cytoplasm localization, we don't know much because it's probably linked to the differentiation of the cells, because EBD is a very highly expressed in differentiated epithelial cells beneath the apical membrane. So maybe when the cells when they start to differentiate, the ABP is not anymore. It's like icatibant, for example. I mean, it's an example, but we don't know much about the trafficking. That it is completely unknown. We don't know much about the trafficking of EBP50 in the cells. We know that it can go from the plasma membrane to the cytoplasm and to the nucleus, but we don't know how it works, how it's regulated. And for the second question is that it's probably maintained, that one of the explanation is that EBP50 maintain, since it binds EGFR, maintain EGFR, stabilized EGFR at the plasma membrane, because EBP50 linked also the actin cytoskeleton beneath the apical membrane. So it's a link that stabilized the EGFR at the plasma membrane. It's what we think. I think it's because, I mean, when you block a pathway here, EGFR, there is compensatory pathway that are, going to be activated. And currently, and she knows that there are other EGFR members that are involved in this pathway, ER2, ER3. I know she works on ER3. And there are currently clinical trials with pan-ER, so targeting EGFR, ER2, and ER4, or ER3. So maybe you need either to target the whole family, or my feeling is that is not going to work if you target just one tyrosine kinase, because you will have ultimately the activation of compensatory pathway. We need to use several targeted therapies. I know that. Well, yes, I have seen that, but I didn't. I know that EGFR can be localized in nucleus and that it can activate some gene expression, but I didn't know why. I mean, I didn't try to understand why. I didn't follow I didn't try to understand, so I don't know why. And I didn't start any studies.
Yeah.
So
in some studies, it is proposed that it can act as a transcription factor, directly,
yes.
or binding to specific response elements in the gene.
Yes, yeah, I know that, but no, I cannot answer to this question because I didn't study anything about that. But maybe it could be interesting to understand the role, the nuclear function of EGFR in tumors, in CCR cells, for sure. No, that's a good question. I know he's very interested by this. No, but if you want to look at the tumor, you are welcome. I can give you the tumor samples and you can check. Yes. Well, we didn't check. I don't remember. We didn't check with Javier the vascularization of the resistant tumor, in fact, because we were very focused on CAF. I don't remember. Maybe Javier remembers something, but I don't think we check. Maybe he checked the CD31 expression, but I think we had. Maybe it's not a good marker for what Luca wants to study, but we didn't, at least by PCR. So we didn't see change, I think. But it's a good point. Yeah, because if you remove the anti-EGFR, even if there are some cells that are already resistant to erlotinib, maybe there are some other that can emerge and they won't have this resistance phenotype again. I mean, still this phenotype. So you have to maintain the inhibition of EGFR. Yes, for sure, because EGFR is so important for the progression. If you don't maintain for future treatment, I think you have to combine. The fact is, you have to combine both to prevent EGFR activation and to prevent compensatory mechanisms.
So thanks very much to all of you. Thank you very much for your attention, and thanks, Laura, to be here. Bye-bye.
Thank you very much to all of you.